This transmembrane receptor is known as a G-protein coupled receptor, indicated by the green clay. When a signaling molecule or ligand represented by the purple clay bonds to the active site of a G-protein linked receptor, the receptor becomes activated. Then it activates the G-protein represented by the blue oval binded to the receptor. The G-protein is indicated to be active because the GDP previously inside has moved out and the molecule GTP has come in. The G-protein then binds to an inactive enzyme, shown by the yellow crescent mold, and makes it active by changing its shape to perform the cellular response. When complete, the GTP is turned back into GDP and the G-protein returns to its receptor. These transmembrane receptors are known as RTKs, or receptor tyrosine kinases, indicated by the long blue fork-like clay molds. When a signaling molecule or ligand attaches to each binding site, the receptors change shape and make contact with one another. This indicates that the receptors are active and ATP molecules represented by the green molds bind to the tyrosines on either side of the receptor. The ATPs then disconnect but leave behind one phosphate group on the tyrosines. Then, relay proteins shown by the red crescent molds bind to the tyrosines and become activated, indicated by the yellow circles on the proteins. When activated, the relay proteins take the phosphate heads and carry out the cellular response, either directly or activating another protein to do so. Morphine affects the G-protein coupled receptor pathway. In the signal transduction pathway to release calcium ions from the endoplasmic reticulum, normally a ligand bonds to the active site of a G-protein linked receptor. The G-protein is then turned on to activate another enzyme, which binds to a protein pump on the endoplasmic reticulum. This pump, indicated by the orange mold, then changes its shape and creates a path for the calcium ions to exit the ER into the cytoplasm. When morphine is present in the G-protein coupled receptor, it binds to the active site of a G-protein linked receptor and prevents the ligand from bonding. Because the ligand doesn't bond, the cellular response can't be initiated, as morphine acts as a barrier. The signal transduction pathway can't sustain, as the reception step of the pathway could never be done. The following slides have symptoms and treatments for this failed pathway.